Let's start our session. We will have today 5 presentations, and it's my pleasure to introduce our first speaker, Valeria Pandea from the Nicolae Destimițeanu State University of Medicine and Pharmacy from Moldova, with presentation role of copper thiosemicarbazone coordination compounds in modulating lipid peroxidation indices in vitro evaluation. Please.
Bună ziua, stimat copreședinte, stimată audiență. Permiteți-mi să-mi prezint prezentarea la tema rolul compușilor coordinativ cupru thiosemicarbazone în modularea indicelor peroxidării lipidelor, o evaluare în vitro. Chimia medicinală se concentrează puternic pe descoperirea de noi agenți antitumorali care sunt atât mai eficienți cât și mai puțin toxici. Proprietățile thiosemicarbazone și complexele lor metalice au demonstrat că sunt potrivite pentru o varietate de aplicații, făcând aceste molecule atractive pentru o evaluare ulterioară ca noi agenți terapeutici potențial în tratamentul cancerului. Administrarea de noi baze și heterociclice și complexele lor de cupru thiosemicarbazone, dar influența lor asupra indicelor peroxidării lipidelor în supernatant în vitro nu a fost studiată. Multe medicamente acceptate în tratamentul cancerului și agenți anticanceroși în elaborarea provoacă efecte secundare severe, care se datorează acțiunilor citotoxice asupra celulelor normale. În ciuda progreselor semnificative realizate în chimioterapia cancerului, toxicitatea sistemică mare și rezistența la chimioterapiile antineoplazice rămân o provocare majoră pentru farmacoterapia oncologică contemporană. Thiosemicarbazonele sunt chelatori ai metalelor de tranziție, cum ar fi cupru, a căror potență anticancer sunt intens investigate. Deși acești compuși din această clasă au fost implicați în studii preclinice, mecanismul lor exact de acțiune este încă necunoscut. În ultimul deceniu, alături de sarcina de a găsi noi medicamente antitumorale, s-a pus problema creșterii selectivității și a reducerii toxicității compușilor noi, precum și crearea de noi strategii privind sinteza unor medicamente selective pentru celulele tumorale. În acest aspect, deosebit de valorat s-a dovedit a fi cercetările științifice privind sinteza dirijată a compușilor coordinative ai metalelor 3D, efectuate la Catedra Chimie Anorganică și Fizică de la Universitatea de Stat din Moldova, sub conducerea profesorului universitar academicianului Aurelian Bulea. A fost obținute un șir de compuși noi metalelor 3D, cu liganzi stelan și macrociclici, ansamblați în urma condensării thiosemicarbazone cu aldehide și cetone, care manifestă proprietăți antitumorale net superioare doxorubricinii, preparat utilizat actualmente pe larg în oncologie. Scopul studiului a fost evaluarea mecanismelor de acțiune a compușilor coordinative ai cuprului cu thiosemicarbazone asupra indicelor peroxidării lipidice în supernatant in vitro de la persoane condiționat sănătoase. În acest slide am prezentat testarea acțiunii compușilor coordinative a cuprului cu thiosemicarbazone in vitro. Primul procesul este de recoltare a sângelor, apoi a fost incubarea materialului, incubatorul cu bioxid de carbon. Mai departe a mers procesul de prelucrare a materialului biologic, păstrarea materialului biologic în congelatorul la minus 40 de grade și testarea indicelor biochimici la spectrofotometru sinergie H1. Cercetarea a fost realizată în vitro pe baza probelor biologice colectate conform principiilor contemporane în standardizarea biologică a experiențelor aprobate de Comitetul de Etic a cercetării al lui SMF Nicolae Destimițeanu, cu emiterea avizului favorabil sub numărul 5 la numărul 38 din 20 iunie 2024 și s-a derulat conform declarației de la Helsinki privind studiul pe subiecții umani, aceștia fiind incluși în cercetare, dar după semnarea acordului informat. Pentru evaluarea particularităților acțiunii compușilor coordinative a cuprului cu thiosemicarbazone a fost efectuate o serie de experiențe in vitro pe șantioanele de sânge periferic colectate de la 10 persoane practic sănătoase. Sângele a fost recoltat dimineața în ajun prin puncte venii cubitale în cantitate de 5 ml. În condiții sterile, sângele a fost introdus într-un flacon ce conținea 20 ml mediu nutritiv egal modificat după Dulbeco, ce conținea heparină, gentamicină și L-glutamină. Pentru studiul influenței remediilor autohtone asupra indicului peroxidării lipidilor, s-a pipetat câte 0,9 ml din acest amestec în godeuri pleci cu 24 de godeuri. În calitate de control pentru stabilirea nivelului inițial al indicilor studiați, în 4 godeuri paralele s-a pipetat către 0,1 ml soluție de ser fiziologic de 0,9% natriu, clorură de sodiu. În celelalte godeuri, acțiunea compușilor coordinative a cuplului cu tiosemicarbazone a fost derivația tiosemicarbazonei ca CMA18, CMD8, MG22, CMC34, CMG33, CMT67, CMG41, T123 și T160, sintetizați la Universitatea de Stat din Moldova în laboratorul materiale avansate în biofarmaceutică și tehnică sub conducerea domnului profesor Aurelian Gulea, testate față de control și de doxorubricina de luată în 0,1 ml ser fiziologic, astfel că concentrația finală se constituie 10 micromol pe litru și 1 micromol pe litru. Toate diluțiile au fost testate în duplicat. Plecile cu godeuri astfel preparate au fost incubate la temperatura 37 de grade Celsius timp de 48 de ore într-un incubator termostatic cu 3,5% de bioxid de carbon. După incubare, conținutul godeurilor a fost transferat în tuburi rependori de 2 ml, centrifugate timp de 5 minute la 3000 rotații pe minute. Supernatantul obținut a fost transferat în tuburi rependori de 1,5 ml, depozitate la temperatura de minus 40 de grade până la determinarea indicelor biochimici. Toate probele au fost codificate. În supernatantul obținut prin micrometode spectrofotometrice au fost evaluați următorii indici ai peroxidării lipidice, metaboliții reactivi a oxigenului, balanța prooxidantă-antioxidantă, produși și proteji de oxidare avansată și de aldehid amalonică. Toate procederile de determinare a indicelor peroxidării lipidice au fost efectuate în laboratorul de biochimie al USMF Nicolae Chistimițanu, după tehnici modificarea noastră, fiind adaptate pentru aplicarea la spectrofotometrul cu microplăci sinergii H1. Aprecierea efectelor compușilor coordinative ai cuplului cu tiosemicarbazone in vitro s-a efectuat conform metodologiei standarde descrise de Râjicova și cu autorii. Evaluarea statistică a datelor obținute a fost realizată cu ajutorul programului calculator statistic SPSS 20 și s-a determinat indicatorii de bază a statisticii descriptive, mediana, abaterea intercortel și procentul medianului. În rezultate, prima figură, unde reprezintă evaluarea in vitro a indicelor metaboliți ai reactivelor metaboliții reactive a oxigenului sub acțiunea compușilor coordinative ai cuplului cu tiosemicarbazone în supernatantul persoanelor practic sănătoase. Evaluând studiul cercetat asupra nivelului metabolitului reactiv al oxigenului al compușilor, s-a observat o majorare a tuturor preparatelor neconcludentă în ambele concentrații în diapazonul de 23-31% față de valorile lotului control. Aici am prezentat analiza mecanismelor de acțiune al compușilor coordonativi ai cuplului cu tiosemicarbazone și efectele lor asupra echilibrului prooxidant și antioxidant în vitro. Influența preparatelor studiate asupra echilibrului prooxidant-antioxidant la administrarea compușilor s-a evidențiat o intensificare reală în ambele concentrații, în doza de 10 micromol pe litru, între limita de 58-130%, pe când în doza de 1 micromol pe litru s-a remarcat cu o majorare nesemnificativă cu 49-122% față de lotul martor. În figura aceasta am prezentat examinarea in vitro a indicului de stres oxidativ produs și protej de oxidare avansată sub influența compușilor coordinative ai cuprului cu tiosemicarbazone. Analiza cercetărilor efectuate pentru produsele protej de oxidare avansată s-a notificat o creștere nesemnificativă la toți compușii în amplie concentrații în diapazonul de 5-11%, neconcludent. În acest studiu, modificările produselor proteji de oxidare avansată și metaboliților reactivi de oxigen s-au dovedit a fi nesugestive pentru procesele peroxidării lipidelor. Acest lucru se datorează faptul că în eforturile de a reduce radicalii liberi se intensifică creșterea balanței pro-oxidante-antioxidante. În această figură am prezentat analiza in vitro a compușilor coordinative a cuprului cu tiosemicarbazone asupra indicelor de aldehidii malonice. Evaluarea acțiunii compușilor de coordinative a cuprului cu tiosemicarbazone asupra indicelui de aldehidii malonice au arătat o scădere nesemnificativă al tuturor preparatelor în concentrație de 1 micromol pe litru, ceea ce indică o intensificare a activității antioxidante. Pe când în concentrație de 10 micromol pe litru se evidențiază o acțiune de majorare veridic a activității pro-oxidante. În discuții, studiul a relevat modularea redox dependentă de concentrație prin compuși de coordonare cupru-tiosemicarbazone, derivații cum este CMT67, au indus o creștere de 2 ori a balanței pro-oxidante la concentrație de 10 micromol pe litru, indicând perioxidarea lipidică crescută și răspunsuri antioxidante compensatorii, în schimb la concentrația de 1 micromol pe litru, CMT67 a redus dealdehid amalonic cu 8%, sugerând activarea antioxidantă. Compușii au prezentat o dualitate similară cum este TIA-123, a crescut de aldehid amalonic cu 25%, dar a scăzut-o cu 24% la concentrația de 1 micromol pe litru, semnificativ, în timp ce CMG41 și TIA-160 au prezentat efecte nesemnificative. Aceste constatări subliniază schimbarea pro-oxidantă-antioxidantă dependentă de doză. Evaluarea in vitro a stresului oxidativ s-a bazat pe produse de oxidare stabile, ca de exemplu de aldehid amalonic, produs și proteji de oxidare avansată, deoarece timpul de înjumătățire scurt al radicalilor liberi necesită tehnici de amprentare pentru a cuantifica interacțiunile specii reactive de oxigen și specii reactive de azot cu biomoleculele. În timp ce modificările produselor proteji de oxidare avansată și metaboliții reactivi a oxigenului nu a fost concludente pentru peroxidarea lipidică. Creșterea balanței pro-oxidante a reflectat eforturile sistemice de neutralizare a speciilor reactive de oxigen și a speciilor reactive de azot. Compușii testați au prezentat efecte sinergice în vitro, mecanisme moleculare rămân neclare, sunt necesare studii suplimentare pentru a elucida potențialul terapeutic, în special în atenuarea daunelor oxidative implicate în boli inflamatorii și degenerative. Acest studiu a constatat că modificările valorilor produselor proteji de oxidare avansată nu s-au dovedit a fi nesugestive din punct de vedere statistic, deoarece valorile au rămas în limitele parametrilor de referință. Acest lucru indică faptul că substanțele studiate nu provoacă sinteză excesivă a radicalilor liberi prosidării la nivel local. În schimb, este posibil ca aceste substanțe să anihileze acțiunea radicalilor liberi datorită proprietăților antioxidante. În rezultatul studiului s-a constatat o acțiune diversă a preparatelor studiate asupra conținutului de aldehid malonic, care reflectă gradul de stres oxidativ. Astfel, produși inovativ ca MG22, CMT67 și TIA-123, în concentrație de 10 micromol pe litru, au o acțiune veridică a activității pro-oxidante, deoarece mărește nivelul funcțional de aldehid amalonic, pe când în concentrația de 1 micromol pe litru manifestă o acțiune antioxidantă, deoarece reduce concludent statistic cantitatea de dialdehid malonic. Concomitent s-a înregistrat o majorare semnificativă a intensității balanței prooxidante-antioxidante, ce ne indică o creștere a proceselor de peroxidare a lipidilor, ce va conduce la intensificarea sistemului antioxidant cu scopul de a neutraliza radicalii liberi. Cercetările actuale asupra proceselor patologice recurg adesea la metode de modelare experimentală a acestora în vitro, obținând avantajul unor explorări la nivel de celule și sisteme enzimatice, fapt ce contribuie la elucidarea mecanismelor patogenice moleculare ale unor maladii și la apariția unor direcții de tratament. Acest studiu a constatat că modificările valorilor produselor proteși de oxidare avansată nu s-au dovedit a fi nesugestive din punct de vedere statistic, deoarece valorile au rămas în limitele parametrilor de referință. Acest lucru indică faptul că substanțele studiate nu provoacă sinteză excesivă a radicalilor liberi prooxidative la nivel local. În schimb, este posibil ca aceste substanțe să anihileze acțiunea radicalilor liberi datorită proprietăților lor antioxidante. În rezultatul studiului s-a constatat o acțiune diversă a preparatelor studiate asupra conținutului de dialdehidă malonică, care reflectă gradul de stres oxidativ. Astfel, produși inovativ ca G22, CMT67 și T123, în concentrație de 10 micromol pe litru, au acțiune veridică activității prooxidante, deoarece mărește nivelul funcțional de dialdehidă malonică, pe când în concentrația de 1 micromol pe litru manifestă o acțiune antioxidantă, deoarece reduce concludent statistic cantitatea de dialdehidă malonică. Concomitent s-a înregistrat o majorare semnificativă a intensificației balanței prooxidante-antioxidante, ce ne indică o creștere a proceselor de peroxidare a lipidelor, ce va conduce la intensificarea sistemului antioxidant cu scopul de a neutraliza radicalii liberi. Cercetările actuale asupra proceselor patologice recurg adesea la metode de modelare experimentală a acestora în vitro, obținând avantajul unor explorări la nivel de celule și sistem enzimatice, fapt ce contribuie la elucidarea mecanismelor patogenice moleculare ale unor maladii și la apariția unor noi direcții de tratament. Preferințele nu o să dau, nu o să le descriu, pur și simplu vă le prezint. Mulțumesc pentru atenție.
Thank you for your presentation. Any question for audience, please? Please.
I'd like to beg my pardon from our Ukrainian friends, colleagues, because presentation was in Romanian. I will also ask the question in Romanian. Pot să vă întreb, vă rog frumos, metabolismul cuprului, deci aveți compuși coordinativ cu cupru. Care este soarta metalului după ce își face acțiunea antitumorală sau așa? Deci ce se întâmplă cu, se adună, se acumulează, se evacuează, se metabolizează?
Păi, n-am studiat în esență cupru, este că trebuia într-adevăr să-l determinăm și atunci lămurim situația clar. Cu timpul o să încercăm să-l studiem mai accentuat ca să știm concret ce acțiune are cupru în celulă, eu știind serul biologic sau materialul biologic.
Thank you. It seems like my presentation will be the next one. I represent a team of biotechnologists from the Institute of Microbiology and Biotechnology of Technical University of Moldova. The title of my first presentation is Modulating Phenolic Compound Synthesis and Antioxidant Activity in Dunaliella salina microalgae by metal oxide nanoparticles under variable salinity conditions. Phenolic compounds are natural antioxidants widely applied for medicine, for food preservation, in cosmetology, and biotechnology is now in the process of searching for the new sources of these compounds. Among the organisms that realized the synthesis of phenolic compounds, microalgae are a very promising source. These bioactive compounds, due to their rapid growth rate, adaptability to extreme conditions, and their very rich biochemical composition of biomass. Donariella salina, one of representatives of a group of microalgae, a green microalga, is extremophile that live in a very high condition of salinity. And have a capacity to accumulate valuable metabolites such as carotenoids, lipids, and also phenolic compounds. The process of synthesis of phenolic compounds in Donariella can be stimulated by different ways, one of which is using of metal oxide nanoparticles, in particularly titanium oxide nanoparticle, zinc oxide nanoparticle, and copper oxide nanoparticle. These three types of metal oxide nanoparticle are important in biotechnology for their properties, redox properties, and their ability to modulate the metabolites in microalgae. The aim of our study was to understand how this three types of nanoparticles can modulate the accumulation of phenolic compounds and the antioxidant capacity of Donariella under a different condition of salinity. We have used two different levels of salinity: low salinity with 60 grams per liter of natrium chloride, and high salinity with 120 grams per liter of salt. Investigating both total phenolic compounds and antioxidant activity in this microalga in this condition help us to distinguish between the effect of oxidative stress and effect of osmotic stress. Nanoparticle used by us was purchased from Aldrich company and was different in their dimension. Our result for total phenolic content. We have three types of compounds and three types of effect of this nanoparticle. In the titanium oxide nanoparticle and the mineral medium with low salinity, we have observed a progressive increase in total phenol content. But in the mineral medium with high salinity, a significant reduction of phenolic content, particularly at high nanoparticle concentration. Zinc oxide nanoparticle, also in mineral medium with low salinity, we observed increase in phenolic content and the gradual decline at higher concentration. The highest stimulation was observed at 0.1 milligram per liter of nanoparticle. In the mineral medium with high salinity, neutral effect at low concentration was observed and decrease of total phenolic content at higher concentration. For copper oxide nanoparticle, the mineral medium with low salt concentration, we observed a consistent simulation of phenol accumulation in all the concentration of nanoparticle applied, but the highest one was in the variant with 1 milligram per liter. in the mineral medium with high salt concentration, stimulatory effect only at the lowest concentration was observed, and not a notable change at higher concentration of this type of nanoparticle. Antioxidant activity of ethanolic extract was also investigated in this case. First of all, I want to notice that there are a big difference between antioxidant activity in the biomass grown in the medium with low salt concentration and with high salt concentration, but all of this, both of these concentration are physiologically comfortable for microalgae. They can grow very well in all these two media. But antioxidant activity is very different. Titanium oxide nanoparticles in the mineral medium with low salt concentration, in this case we not observed some significant effect on antioxidant activity, and the mineral medium with high salt concentration and stimulatory effect was observed only in highest concentration, 30 milligram per liter. For zinc oxide nanoparticle in medium with low salt concentration, we observed a decrease of phenolic compound in concentration of 20 and 30 milligram per liter, and in mineral medium with high concentration in the same two variants we observed a stimulatory effect on antioxidant activity. Copper oxide nanoparticle in the mineral medium with low salt concentration, at the concentration of 10 and 20 milligram per liter respectively, was observed a decrease in antioxidant activity and an increase of active antioxidant activity in the highest concentration of nanoparticle used. In the mineral medium with high salt concentration was observed some variation, but not significant variation of antioxidant activity. We tried to summarize the observed effect on microalgae. And for phenolic content, depending on the combination of level of salinity and the concentration of nanoparticle, we have observed three types of responses. Stimulation of phenolic synthesis, which was observed in the combination of moderate salinity and low nanoparticle concentration for all three types of this nanoparticle investigated, and in condition of high salinity and high nanoparticle concentration, the inhibition of phenol photosynthesis in Donariella salina. In two different combinations, moderate salinity, high nanoparticle, and high salinity, low concentration of nanoparticle, we observe a divergent response typical for each type of nanoparticle separately. Also for antioxidant activity, in this case, also three types of responses was noticed. In case of moderate salinity and low nanoparticle concentration, high salinity and low nanoparticle concentration, we was not observed has some significant effect on microalgae. It seems like low nanoparticle concentration is the main factors in this response. In the case of combination between moderate salinity and high nanoparticle concentration, we observed a divergent response depending on the type and dose of nanoparticle. No significant changes for titanium oxide nanoparticle, reduced activity for zinc oxide nanoparticle, and sharp increase in antioxidant activity in the case of copper oxide nanoparticle in highest concentration. Our conclusion are the next. The interaction between titanium oxide, zinc oxide and copper oxide nanoparticle and Dunaliella salina is influenced by both the nanoparticle type concentration and the salinity of the culture medium. Low nanoparticle concentration and moderate salinity enhance phenolic synthesis, with copper oxide nanoparticle showing the most consistent stimulatory response. Osmotic and oxidative stress induced by salinity, high level of salinity and the high nanoparticle concentration inhibit the production of phenols and triggers the enzymatic antioxidant defense mechanisms. The data presented underscore the importance of modulating nanoparticle application according to specific environmental conditions to optimize biochemical response in microalgae system. This work is a part of biggest one, which include wide variety of nanoparticles and a lot of microalgae and cyanobacteria. We try to understand how nanoparticle can be used as biotechnological tools in modulation the production of bioactive compounds by these valuable biotechnological objects. Thank you. Now question.
Yeah, I have a question. So in this study, your main focus was phenolic compounds from the microalgae, correct?
Yes.
And these were like the compounds that produced the antioxidative effect, right?
Yes.
What about the other compounds that are produced by algae? Because in the first slide you mentioned, or one of the first slides, that there are like other things produced like lipids and something else. Yeah.
Yes,
What about
we
those
have
compounds?
these results, but for this presentation was found only phenolic compounds and antioxidant activity. Antioxidant activity is not determined only by phenolic compounds, and you can see that the response of phenolic compounds and antioxidant activity are different. We have this result. We have many publication in the synthesis of pigments, proteins, lipids, carbohydrates, and other compounds which are for interest, for biotechnological interest. In this work we focused only at it.
So what I mean, your conditions, maybe they stimulated or inhibited production of those other compounds. So maybe there would be something interesting to see.
The effect produced by nanoparticle and other compounds depend on level of stress induced to the object. In condition of low level of stress and the medium level of stress, you can use this product for biotechnological purposes, because the accumulation of radicals in the biomass is low and the quality of biomass remain at a very high level. But in the condition of high level of oxidative stress, which is determined by high concentration of nanoparticles in our cases, produce accumulation of product of peroxidation, as in previous presentation was, and this biomass can be used for human or animal use, or for producing some pharmaceutical or nutraceutical. So for each type of stimulators, you have to we have to find the best condition to ensure the accumulation of product of interest and the quality of biomass. And control of quality is very important and strictly necessary in biotechnological production using biostimulators.
Okay, thank you.
Thank you. And the next presentation is also mine, because our colleagues from Armenia aren't here, and that's why. This work only uses the nanoparticle as the object of study, but we investigated the effect on the plants, in this case of calendula. The title of my presentation is Foliar Uptake and Translocation of PVP-coated metal nanoparticle of copper, gold and silver in Calendula officinalis. Nanoparticles have become integral to modern biotechnology and are increasingly released into the environmental through industry, through agricultural formulation, and by the natural causes. On this process, they inevitably interact with plant and can produce some changes in the composition of plant biomass, and also can be accumulated in the plant because they can penetrate through the different structure and can be translocated from the leaves to other part of plants. Understanding how nanoparticles penetrate, accumulate, and influence plant metabolism is crucial to predict their long-term ecological impact and to design a safe, efficient application of different type of nanoparticle, especially in agriculture. Calendula officinalis serve as a suitable model for such study due to its known antioxidant system and sensibility to oxidative stress, and investigating the response to metal nanoparticle provide insight into the balance between beneficial and adverse physiological effects, helping to define the threshold between biostimulation and toxicity in plant nanomaterial accumulation. Copper, silver, and gold nanoparticle were selected for their distinct redox property and for its use in very different area of human activity. Especially this type of nanoparticle are now present in lotion, in cosmetical product, in medicines, and in biofertilizer and biopesticide due to their antibacterial, antifungal, antiviral properties and other. The aim of this study was to evaluate the response of Calendula officinalis to foliar application of gold, silver, and copper nanoparticle, to determine the effect of different concentration of this type of nanoparticle solution on the biochemical parameter and antioxidant activity in leaves, and to estimate the ability of Calendula to accumulate and translocate three type of metal nanoparticle from leaves to other segment of the plant. Calendula were exposed to three type of nanoparticles under foliar spraying condition in concentration from 1 to 100 of milligram per liter every 2 days for 28 days. The starting of spraying was in the 60th day of cultivation until the flowering period. And in each plant received 2 milliliter of nanoparticle of different concentration solution per one application. After 28 days of application of nanoparticle, plant material were collected for analysis. Plant material was divided in leaves, stems, roots, and flower, and the analysis of accumulation of metal in this part of plant was made separately. During the spraying, the soil was covered with cardboard to avoid the contamination of the soil with nanoparticle and absorption of this nanoparticle from the soil by root. So we can be sure that presence of metal in the roots is due to the translocation from the leaves and not by accumulation from the soil. This is our nanoparticle. Nanoparticle was made was synthesized by the physician, so it was a necessity to study and determine the dimension of this nanoparticle. Our nanoparticle was from 1 to 5 nanometers, silver nanoparticles 1 and 2 nanometer, and copper nanoparticles was bigger from 15 to 17 nanometer. This slide include two type of results. In the first part, there are the accumulation of metal in different parts of plants, root, stems, leaves, and flower. And we will follow one column for one slide. In this column, we follow orange slide in the first part of the slide. Accumulation of gold in the part of plant. It's worth to mention that gold isn't present in control plant. It's not typical for the plant to have gold in the root, stem, leaves, or other part of the plant. The highest gold content was determined in calendula leaves and stems, which was sprayed with 100 milligram per liter of gold nanoparticle. And gold was found in the root at all applied concentration. This type of nanoparticle caused reduction in carotenoids and the chlorophyll B content in all tested concentration, and also low concentration from 1 to 5 milliliter per liter increased the content of chlorophyll A, whereas higher concentration lead to a decrease, evident decrease in this pigment quantity. An increase in antioxidant activity was observed at all tested concentration of nanoparticle. In this slide, in the first part, we will follow the gray column, the concentration of silver in the different part of plants. Also, as gold, silver was not present in the plant in the control group, but this type of nanoparticle inhibits the process of bud formation and flowering of calendula under 15 and under 100 milligram per liter. No flower were formed with this plant. Also, this type of nanoparticle caused a decrease in the amount of chlorophyll B and carotene in all concentration applied to plant, and of chlorophyll A only in high concentration. Silver nanoparticle also generally induced an increase in antioxidant activity, but phenolic content significantly increased in low concentration and decreased compared to control at a higher concentration of nanoparticle. In this slide, in first part of the slide, we will follow the green column. Copper content, copper is present in control plants, and copper content in roots decrease compared to control in the variant of application of nanoparticle to plant by spraying. An increase in copper content in leaves was observed under 1 to 50 milligram per liter, and a decrease at highest concentration of nanoparticle. For copper nanoparticle, antioxidant activity in the leaves showed significant changes only in concentration of 50 and 100 milligram per liter. And the phenolic content decrease at low concentration and remain unchanged at higher concentration relative to control plants. So we observe a difference in metal accumulation in the aerial part of plants. We observe the highest accumulation characteristic for gold nanoparticle in stem and leaves of Calendula officinalis. An increase in the concentration of copper oxide nanoparticle reduced in the decrease of copper, result in the decrease of copper content in the roots compared to control. Plant treatment with gold and silver nanoparticle lead to an increase of their content in roots, most probably due to root secretion into the rhizosphere part of the soil. Low concentration tend to stimulate or have no effect, while higher concentration often reduce pigment level and alter antioxidant and phenolic profiles. Gold and silver nanoparticle generally enhanced antioxidant activity, whereas copper showed a concentration-dependent response. These results are also part of big investigation of the influence of nanoparticle applied by spraying and by watering, and we have also the result of nanoparticle accumulation in soil and the influence of this nanoparticle on activity of microbiota present in the soil during the experiment with watering, and also in the experiment with spraying, because one of the mechanisms of detoxification is the elimination of the toxic metal, and they are eliminated mainly by roots in the rhizosphere zone and affect the activity of microbiota in this site. Thank you. And question, please.
Yeah, a question. So you used simultaneously 3 types of particles, right?
Not simultaneously, separately.
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 So when we see like copper nanoparticle concentration, we see that this is only these plants were only this type of nanoparticle was applied to it, correct?
Only these, yes.
And do you have an idea what is going to happen like in the next step? For example, now you treat plant with nanoparticles, you see an increase in concentration. But what's going to happen in ecosystem when this plant is consumed by, let's say, an animal? Is it possible to model that, like what's going to happen? Or maybe these nanoparticles will go to soil, or maybe into the air again, or maybe they will go to like animals that consume the plant?
As plant as the first player in the food chain, and the highest level in food chain concentrate all the type of xenobiotics. We think that this phenomenon of accumulation of nanoparticle in the part of plant, it's very dangerous, and we have long-term evaluation of these processes, because they rest in the flower and leaves, and flower and leaves are used for infusion or for preparation of the creams or other product, and can be part of food chain, and not only food chain, but are used by human and animals, and can be dangerous for all the biosphere, not only for plants. We demonstrated that nanoparticle can be transferred from, in case of watering of plants, in the tea obtained from the flower. And the concentration factor is low because the concentration of nanoparticle in the environmental now is low, but with widely use of this nanomaterial, it will become dangerous.
So maybe it's a good idea to maybe even select some kind of cosmetic product and see if you begin to see these nanoparticles in those cosmetic products.
In cosmetics, there are another problem. The nanoparticles are introduced as anti-aging or as antibacterials. So the widely use of different cosmetic products with nanoparticle, I think, will have effects in long term.
So it's they can accumulate in the body, and then after 10-15 years.
Also, our group have had the investigation of accumulation of biofunctionalized nanoparticle in different organ of animals. And this little, very small nanoparticle can pass blood-brain barrier and are accumulated in brain. And we investigated 28 days of clearance, and gold and silver nanoparticle remain in brain in the level of 70%.
Yeah,
it can be very dangerous. That's why we need a solid quality control in biotechnology.
Thank you.
Thank you.
Have you considered other particles? Thank you. It's a very nice study of these, well, cosmetically used nanoparticles. But have you considered in the high concentrations that is outside the particles which are formed naturally, for example, like magnetic nanoparticles from the iron oxide? Because they are more, let's say, at concentrations you have been testing these ones, it's more appropriate for concentrations of the much more produced nanoparticles in the industry.
Of course, it is very interesting to study all type of nanoparticles that are in very big concentration in the environmental. But our group work now with 5 type of nanoparticle. We are only 7 researcher, so we can do all we think to do. But we'll in the future, maybe. Thank you. Our next speaker will be Razvan Gerasim.
So, thank you very much for introduction, and good morning everyone. My name is Razvan Gerasim. Currently I'm a research assistant at the Petroponi Institute of Macromolecular Chemistry from Iași, Romania, and in the next few minutes I would like to share with you a part of my field of research. So, in collaboration with a Chinese chemical company, we developed a few copolymers based on 2 different polymers. One polymer is represented by polyhistidine, and the other polyhistidine is represented by hydrophobic part, and another polymer is represented by polyethylene glycol. Of course, it's a hydrophilic part. With this company, we developed 3 copolymers with the same length of polyethylene glycol, and polyhistidine with a different units of histidine, 20 units, 26 units, and respectively 31 units. These 3 copolymers, in the first study, we're assembling an unloaded micelle. Micelle are spherical nanoparticles in a nanometric domain. This assembling, it's possible in pH 7.4, and of course it's possible to incorporate a different compounds. For example, in this study we use the doxorubicin for models, and it's possible, of course, to assembling in doxorubicin assembling in the hydrophobic part of the micelles. In terms of the size, one of the techniques used is scanning transmission electron microscopy. In the left side, we can observe the stem images represented by different bispherical micelles. You can see here we have two different regions. The inside region is with a high intensity, represented by polyhistidine due to the high density of the polyhistidine, and of course the shell represented by polyethylene glycol. And here, in the next three stem images, we can observe the increase of the intensity of the micelles. This increase of this intensity is due to the presence of the doxorubicin molecules inside inside micelles. So these micelles, we developed these micelles for pH sensitivity. To determine these parameters, we use the release studies of doxorubicin. So for this experiment, we use three different pHs: 7.4, 7.4, and respectively 6.5 at 37 degrees Celsius. You can see here in the all of curve release, we use, for example, for control, we use the free doxorubicin with the pink curve. And you can see here, for example, we have a low release after 10 hours. Here, with degrees pH, we can observe the release it's increasing. And of course, in the final pH 7.5, we can observe after 10 hours, approximately all samples, we release approximately 70% of release of doxorubicin. In the next part, we use the same copolymers, but with different functionalizing group on the terminus of the different polymers. For example, in the first one, we use the end of the polyethylene glycol to functionalize with a linker based on maleimide, and the other one, it's represented by functionalization of the terminus of the polyhistidine with rhodamine B. To determine this functionalize, we use the mass spectrometry in this part of physical chemical characterization. Sorry. Okay, in the next part, we have three different solutions. For example, here we use the copolymers with the simple copolymers and the copolymers functionalized with maleimide on the end of the polyethylene glycol chain. We're auto-assembling in the unloaded micelles, and of course here we have the copolymers with maleimide and the rhodamine. We're assembling in the micelles loaded with rhodamine. And of course, finally, we use the doxorubicin to incorporate the physical incorporation in the micelles with the linker on the surface of micelles. In the final part, to functionalize the surface of the micelles, you use the trastuzumab. Trastuzumab is one of the most used drug for breast cancer. In this chemical part, we modified the all free amino group on the structure of trastuzumab in thiol group with the specific agent called Trout reagent. And then, for example, the thiol imine group reaction with the maleimide group on the surface, it's 100% specific reaction. And then finally, we obtain three different micelles. For example, the unloaded one with trastuzumab on the surface, the rhodamine, and respectively the doxorubicin inside with trastuzumab on the surface. In terms of the size, the first technique was dynamic light scattering. With dynamic light scattering, it's possible to obtain the hydrodynamic diameters. We can observe here, for example, the unloaded micelles. We have these hydrodynamic diameters. After the attach the molecule trastuzumab on the surface, the diameter it's increased, significantly increased, of course. With this technique, it's possible to obtain the zeta potential. Zeta potential, it's with this this parameter, it's possible to observe the different modification on the surface. In the next part, about the quantification of the bioactive molecules. For the samples with rhodamine, I use the fluorescence spectroscopy, and for the doxorubicin, I use ultraviolet visible spectroscopy. For both, I use the calibration curve method. So, in the next part, I use, for example, I use different solution of the micelles. So basically, this experiment is to demonstrate how to influence the biological compound inside of the micelle. For example, here you use the 6 different micelle solution. With 10,000-fold dilution below CMC. CMC, it's called critical micelle concentration. Critical micelle concentration is one of the important parameter in moment we need to work with micelles. And it indicates what is the minimum concentration when you use to make the micelles. So here in the first one we have the unloaded micelles. With 1000-fold dilution after 15 minutes and after 62 hours. For example, here you use the nanoparticle traking analysis methods. This method you use to obtain the Brownian motion of the nanoparticles. And of course, it's possible to obtain the hydrodynamic diameters with this technique. Here we have the unloaded micelles after 15 minutes of dilution at 10,000-fold. And here we have the same sample after 72 hours. Here you can observe a very different information about these samples. You can conclude in here, after 24 hours, the unloaded micelle is totally disassembling in a solution. The same information you can observe here in case of unloaded micelles, but with trastuzumab on the surface of the micelles. Here we have the after 15 minutes, and of course in the final image NTA video, we can observe a slightly different compared with this one. The next one, it's represented by samples with rhodamine. Here we have a little different compared with unloaded micelles. For example, here we have micelles loaded with rhodamine after 15 minutes, and of course after 72 hours. And here we have the micelles with rhodamine and with trastuzumab on the surface. We have the approximately the same result. Here we're concluding the rhodamine influenced the stability of the micelles after dilution below CMC, and of course after a different time. And in the final part, here we have the sample with doxorubicin, doxorubicin without trastuzumab, and here we have doxorubicin with trastuzumab on the surface. Here we don't have the different significant. Okay, in terms of the therapeutic effect, in this part we use two different cell. The both is represented by a cell of breast cancer. The first one, it's SQBR3. It's represented by overexpressing specific receptor for trastuzumab called HER2. Receptor HER2, it's epidermal growth factor type 2. And the other one, it's B24744. It's a tumor breast cancer line with normal expression of HER2 receptor. Here we have the two graphs of cytotoxicity at the different samples at the different concentration. For example, here we have the control, we have free micelles. And I used two different concentration of trastuzumab, 4 micrograms and 8 micrograms, and the same concentration in the functionalization on the surface of the micelles. You can observe here we have a very interesting differentiation between free trastuzumab compared with trastuzumab functionalized on the micelles. This is the same result you can observe in the other one type of tumor cells, but here the effect is very, very high compared with this one. Okay, and I would like to thank you very much for your attention, and if you have questions, please. Thank you.
Thank you for your presentation. Any question or comments from audience?
About this time-based evaluation, what kind of a solution did you place these functionalized compounds in? Is it like blood, or is it just some kind of a neutral solution?
The all functionalization, it's in the buffer solution. It's in all the all functionalization. It was in buffer solution in 7.4 and 7.6 pH.
So it's interesting. What's going to happen if you place it? If you try to place it in a more like natural solution, such as human blood, and see what happens in 72 hours, for example, how stable it will be. Maybe there is gonna be like a difference. Who knows? Yeah, because it's gonna be more like similar to real environment where the drug will be.
Yes, but here in the drug release study, I used three different pH.
Yeah.
For example, 7.2, it's like a pH in extracellular extracellular tumor or pH. And for example, 7.5, I think it's in approximately the center of the tumor cell.
So this it's like we are simulating that. Let's say this solution is ended up inside this tumor, and now we are looking at what's going to happen like over time. Correct?
Correct.
Okay, and maybe a question about these polymers used. So I understand that you use a polymer, like a container for a drug, right? So that it will not release.
Yes, for us. Just a second here. Yes, for example, the polyhistidine. When you work with, for example, the basic solutions, these polymers, it's deprotonated. After deprotonating, these polymers, it's auto-assembling in a different structure. And to obtain the micelles, it's mandatory to attach a different polymers with a different compartment. For example, the polyethylene glycol, it's hydrophilic part, and it's very important to, for example, the micelle is very important to have two different parts, the hydrophobic part and the hydrophilic part.
So it's like it's both attracted and rejected from water, right? From water molecules.
Yes, because here it's the auto-assembling of this copolymer in micelle, it's in aqua solution. Here in aqua solution.
Interesting. And do you know what is going to happen after the end of life cycle of these molecules? So, for example, the drug is released eventually, and then the polymer is still in the body.
Yes, but this polymer it's biocompatible, 100% biocompatible with normal cell. Yes, it's not toxic.
Yeah. Okay. Thank you.
Welcome.
Other question? Thank you.
Thank you very much.
Our next speaker is Alexandru Rotaru from Petroponi Institute of Macromolecular Chemistry, Romania.
Well, you. Thank you for the kind introduction. My name is Alexander Rotar. I'm coming from the same institution like previous speaker. And my talk will be about the hydrogels, and not about the commercial polymer-based hydrogels, but about the hydrogels based on the small molecule and on the processes of self-assembly, kind of supramolecular chemistry, and the formation of smart materials with the small molecules. And the hydrogels we work together is based on the molecule called guanosine, and guanosine is one of the 4 canonical bases which DNA is made of. In a normal double-stranded DNA, guanosine is forming three hydrogen-specific bonds with cytosine. But when the DNA in a human body is guanosine-rich, it is possible that guanosine participates in some different supramolecular assemblies, like these guanosine quartets. They are not very stable unless they have the metal ion inside, and not any metal, but specifically metals which fit inside these quartets. And these are typically potassium in the human body, but outside the human body it could be strontium, it could be barium, or any other metal which is able to fit inside the cavity and able to form the coordination bonding and stabilize the quartets. And but we will not talk about molecular biology because we are chemists. We are interested more in molecular structures. And this guanosine molecule I was talking about is a commercially available product. It's quite cheap, so one can just buy it and use it for different kind of chemistries. And scientists indeed so far have synthesized hydrogels based on guanosine, like it's shown over here. But the problem is that guanosine molecule itself, it's not very soluble in water. And the hydrogels they are obtained, they are let's say not very transparent, they are very fragile, their properties are quite bad. And it's because the guanosine quartet is tend to stack like in nature. This is kind of similar to nature structures, but since we have individual molecules, this quartet is just stacking together, forming this jellified structure, but it's not transparent due to the water solubility problems and all other issues. So we decided to improve the system, and we observed that guanosine is having these 1,2 diols, and these 1,2 diols specifically participate in the reaction with the boric or boronic acids, forming these boronic esters in between. So if we use 1,4-diboronic phenyldiboronic acid, it would work like as a linker, which will bind two guanosine moieties, and now we have a dimer. And imagine that each of the guanosine over here is forming a quartet over here, and then it means that it will form a stretched plane of guanosines. And since they like to stack, they will form also stacking, so it will be a 3-dimensional network. And since it is all taking place in water, the water will be trapped inside this network. And then we will obtain, as we obtained actually, a very nice transparent hydrogel. This is a typical test we are doing. It's a vial inverted test. So the gel is formed, and then we turn off the vial upside down, and we see that if it stays on the top, then the gel is quite firm. And this test is just a provisionary test because all the gels afterwards go to the serological studies. But so far we were happy that we could obtain using this method, and it was easy, the gel was transparent, and it can be used for various applications. And of course, the first thing we tried is this gel over here was formed due to the presence of the potassium ion inside the reaction mixture. But we then adopted this method using different ions also. For example, barium, which also fits in here as a central atom. And we observed that barium atom actually participated in some different binding mechanisms and allowed for the accumulation of much more water inside the gel matrix than the one with the potassium. You see the comparison of the volumes of what it can hold. But then the surprise came when we used the magnesium ions. But the magnesium ions was just a mistake. We wanted to wash the potassium hydrogel in a buffer containing magnesium, and overnight we could see that the hydrogel just swallowed, took the whole volume of the hydrogel, and we observed that it could hold and withstand the volume of 44 milliliters, which is like 15 times much more volume than the initial hydrogel. We suppose that the magnesium ions, they do not form the quadruplexes because it's not possible, but they participate rather in interplane connections of the network. Well, what we did next is, of course, try the check somehow the mechanism of assembly inside. We used atomic force microscopy, and it was obvious that all the investigated gels had a different internal structure. For example, the potassium gel formed this wave-like micrometer-sized waves. The barium-based hydrogel was a more amorph one, and the magnesium hydrogel formed these wire-like structures inside the hydrogel. Next step was, of course, since it was beautiful material, transparent material, it could be used for biomedical application, but prior to biomedical application, we have to test its toxicity. And since already the results of this first generation hydrogel was quite bad, the potassium hydrogel gave a cell viability of fibroblasts we tested around 40%, which is not very good. Well, the barium hydrogel gave slightly better because it has more water inside, and the only one which was suited for biomedical application was the magnesium-based hydrogel. And you can see also here the test, we tried to deposit some cells on the surface of hydrogel, but the morphology tells us that these are mostly dead cells. Well, in case of barium, they are more or less dead and alive. Magnesium was much better, of course much better, but the huge amount of water inside the matrix made this material quite fragile and very difficult to work with. So this first generation of hydrogels we have obtained told us that in fact the water content is important. So we decided in the second part and second generation of hydrogels, we decided to improve the water content of the hydrogel by providing some composite ones. So we decided to use the carbon nanotubes, which are extremely beautiful materials for enforcing any kind of matrices. But native carbon nanotubes, they form these bundles. They hold together and they are very difficult to open. So if they open for a short time and then they tend to aggregate. So what we did is we decided to use them because we think that this molecule has quite enough non-polar pieces of the molecule which can actually stabilize the carbon nanotubes. So in fact what we did was we adapted the synthesis. We used instead of potassium to obtain this dimer, instead of potassium we used lithium, which doesn't form the quartets. So we obtained this dimer, then we mixed dimer with the carbon nanotubes and sonicated it for half an hour. And when the carbon nanotubes began to disperse, the matrix of the gel wrapped around the nanotubes and stabilized them. And then subsequent addition of potassium ions led to the sealing and, let's say, reticulation of the system. We could obtain, of course, the hydrogel here, a carbon-based hydrogel. And comparing to the same amount of water, you see that this one is collapsed, but this one is actually intact. Then we investigated the amount of carbon nanotubes we can add to the hydrogel, and we could see that the water content of the hydrogel is strongly dependent on the amount of the added carbon nanotubes. But the problem is that carbon nanotubes also are toxic, and we found different ratios of water carbon nanotubes to tune the toxicity, and we actually reached the toxicity which is okay for the biomedical application. We tried to deposit some cells. Cells were feeling pretty okay, so we considered this second generation hydrogel was okay for cell support application. But the problem is that the color, black color, is not very suitable for the optical investigation of the biological tissues. So yes, it's okay, but still have some disadvantages. Then we decided to make the third generation of hydrogels and to apply some, let's say, polymers, biologically, let's say, compatible polymers, and we adapted again the chemistry. So what we did is we didn't obtain in the first step the dimer, we obtained only one modification of the guanosine on the linker, but the second moiety of the boronic acid we left open to react it to a template, a biologically adapted template. And we decided to use dextran. Dextran is a nice biopolymer, biodegradable, non-toxic, and easily available. And the thing is that dextran has 1,2 and 1,3 diols, which can easily react over here. So it means that we can graft various amounts of guanosine on the template of dextran, like is present here, and we can tune the properties of the hydrogel depending on the amount of the guanosines we attach to the template. We did that. So this is schematic presentation. Then at the last step we add potassium, which fortified the structure, and we could obtain nice and beautiful transparent hydrogels based on the dextran template. We tried to cover all the diols as a sample, sample 1. Sample 2 we tried to cover only half of the diols, and sample 3 we used 30%. But all three ratios between guanosine and polymer yielded us nice and transparent gels. Toxicity was more or less okay on the gel 3 and 2. Gel 1, where all the guanosines were substituted, the diols were quite toxic one. And here you can see the morphology of the cells. This is the one which was toxic one, but still cells adhere to the surface. They feel quite good due to the morphology you can see. So we consider this hydrogel is the best optimum variant we could obtain with this system. Well, what is next? Since we had nice results with this diol and dextran molecule, we decided to use another oligosaccharide called cyclodextrin. A very interesting molecule, just a couple of words about it. It's a barrel-like molecule which is structured in such a way that interior of the molecule is hydrophobic and exterior of the molecule is hydrophilic. It means that if geometrically a molecule can fit inside, it's not soluble in water, the cyclodextrin can solubilize it in water. It's a very nice property, and this property is very often used in cosmetics and food chemistry. Well, there are three types of cyclodextrin, alpha, beta, gamma, and all are different by the numbers of sugar moieties inside the circle. But the interesting thing that on the bottom of the cyclodextrin we have these pairs of diols which are suited for our chemistry to make some interesting gels. And that's what we did. We used these 7 pairs of diols, reacted them with a linker, obtained 7 linkers attached to the bottom of the cyclodextrin, then added the guanosine. So this one, in the presence of potassium, could potentially form this kind of a network, which is completely random. But as quartets like to stack, cyclodextrin also like to stack. So we finally could obtain the structure which we think, and our results show us that it looks like something like that. And different amounts of guanosine in the attachment to the cyclodextrin give also different transparent hydrogels, but these gels showed extremely high toxicity when we tested them on cells. Why? Because cyclodextrin has a very strong property and affinity to cholesterol, so it actually attracts cholesterol from the cell membrane and thus destroying it completely. But on the other hand, if this property is very nice, it can be used as antibacterial agent. So we tested our system for various antibacterial strains, and we found that Staphylococcus aureus actually was selectively destroyed both in planktonic form and in biofilm. So it actually represents a very nice and interesting approach for antibacterial testing properties of the materials. One of the interesting things about these hydrogels is the fact that all hydrogels I mentioned above is the fact that this, I hope it's working, this video. No. Yes, this hydrogel had very nice property called thixotropicity. It means that during the mechanical shaking they destroyed the hydrogen bonding, they became liquid, and then if it's left to rest it can become back solid. So it also very correlated much with the property of injectability. So these kinds of gels can be easily loaded in a syringe and then injected, and if they're injected properly in a liquid they maintain their shape. As you can see here, the worm-like shape from the injection is maintained. So it means that all this hydrogel I mentioned above, they are suitable for 3D printing of organs, for example, if properly tuned. Next system, we decided to induce stimuli responsiveness to the system, and particularly we would like to use light to improve the antibacterial properties of the hydrogel. So again, we use these cyclodextrin-based hydrogels, but then we introduced 2 nanomaterials. One is graphene oxide and the other one is carbon nanotubes. We adapted the system. We obtained several series of hydrogels with different concentrations. Why is that? Because during irradiation with a laser, particularly red laser, these materials are able to heat and they're able to kill much better and to have effect of antibacterial properties. And here is an image of testing. The first row is a hydrogel without any carbon material, and you see that during 10 minutes of radiation There is no temperature increase in it, so it's a kind of negative control. The sample with graphene oxide showed an increase, but not high, so approximately to 30 degrees. And then the sample with the carbon nanotubes, in fact, gave a huge increase of temperature up to approximately 48 degrees. And this graph shows that this process is cyclic, so the system is stable. This is a heating, cooling of the system, and it shows almost repetitive results. Next, what we did is we tried this on the same Staphylococcus aureus strains, and indeed the radiated versus non-radiated samples. Even so, the heating of the carbon of the graphene oxide was not very strong. Still, it gave a difference between the radiated one and the irradiated one. I think this is because different mechanism of action also happened, like for example, reactive oxygen species formation and maybe other. But generally, the system was much better improved with nanomaterials rather than without them. What we did next is also we used this in another system. We used the cyclodextrin to encapsulate antibiotic, or let's say antibiotic agent, and we took this. Before that, we adapted it. We left several of the boronic acid moieties free, so this system can bind to the surface of the bacteria, because surface of bacteria has a lot of sugars and of course 1,2-diols. So to be very specifically, if they catch this cargo, and then we studied also the properties. It was very different depending on how many guanosines are attached to the bottom of the cyclodextrin. And then we applied the same system to the drug-loaded cyclodextrin. So first we loaded the norfloxacin inside the cyclodextrin, added the linker, added the guanosine, yielded the norfloxacin-loaded hydrogel, and then tested it for bacterial strains. And yes, a gel without norfloxacin was less potent than the one with the norfloxacin, so it means that the system is working. And more importantly, the kinetics of the release of the norfloxacin is much slower. It means that the properties of antibacterial can be prolonged for much more time. I think this was the last slide I would like to show, and finally I would like to thank my colleagues for the helping obtaining the results you have just seen, and finally to thank the project who helped us to be here with you. Thank you very much.
Thank you for your very interesting presentation. Any questions from the audience?
Yes.
My question is not about your scientific things. Thank you, it was very interesting. My question about where you have the opportunity to discuss your results with person who could use this in their business, in their life, in their different industry purposes.
Thank you. It's a technology transfer question.
Oh, well.
Yes, approximately. Well, the study you have seen, just a small answer is that we have an interdisciplinary center, and we can test them. So results you've seen, they tested in our lab. But then we have collaborations, and one of the major collaborators of ours is the Regional Oncological Institute. So we always discuss with them all the system we obtained, not about this one, but also the system from the previous speaker. So all the system we obtained, we try to discuss with the real doctors from the real hospitals, and they help us to understand their needs. And then as soon as they say, well, we need a system which releases the drug slower, then it goes without any incorporation, let's say. And then we say, well, we have this kind of system and this kind of system, and then they correct us. They say, well, this one is not good because it will accumulate in tissues and it will not metabolite, and we don't like it. But then this one, oh, this looks okay, so we'd like to try it. We'd like to give it a deeper investigation, because we are limited, but they are not. They have even human tissue accessible for testing. So this short question, answer to your question, yes, we collaborate with the real hospitals, and they tune our research towards the practical application.
And is it significant approach, for example, in conference, to tell about this type of collaboration, or not, or it's not an interesting question for conference, for example.
We are fundamental researchers, so our research is fundamental. I think I don't see a practical application in a long term, because you know that any medical or any system for biomedical application needs a lot of testing, lots of finances. So we actually disseminate our results easily without being afraid that somebody will steal it, because we actually publish them first, then we disseminate it. So we don't see any unpublished results here. But generally we never had any situation where we had a conflict of somebody, let's say, harming our results and saying that we can steal them and use them. No.
Thank you.
And about this, so you showed us like 4 or 5 generations of different hydrogels. Do you already have like an idea where you can apply them with these medical people? Let's say, because I saw that first, second generation, maybe those would not be applicable, but some of the latter, let's say hydrogels, especially the one with high cytotoxicity, probably is applicable in some situations, right?
Yeah. Thank you. Nice question. Indeed, yes. The short answer is no. They didn't work in real life. We went with some samples to an institution which printed organs. And we proposed them some of the best candidates, and they tried. They have also preliminary results, and they say, well, it's working or it's not working. And unfortunately, we didn't pass the first step. So they said that the material was nice. We had like 8 hours of discussions, but then we went to the lab, and I stand by the operator, and he said that no, it's not holding the shape as it should be. And then they said that yeah, come with a better candidate. And I think we kept this part of organ printing. But other than that, the cancer hospital didn't like this system at all. Yeah, so we're still looking for others who may be more interested in this research. But for us, it's not so important, because we can still publish them. These are these points we have to disseminate for the projects, and since we are fundamental researchers, it's okay for us. If they will be applied, okay. If no, not okay, but we can live with it.
Yeah, but still great to get this feedback, so that you know, like, okay, this maybe we should move in this direction a bit, or maybe modify this polymer to get better result, and so on. Yeah,
That's true.
Thank you.
Other question? No? Thank you.
Thank you.
Now I would like to thank all you, the speaker, for a very interesting presentation, and the audience for curiosity and for interesting question that made our session interesting and interactive. Thank you.
Спасибо.